Hello, welcome back to part 5 of our look at blood. This will be the final video that we will have covering the information for um, blood. And so what we're going to focus on here is blood typing, which is something that you've spent a good amount of time looking at in lab. But we're going to go ahead and just kind of make sure that everybody is good and clear on um, what blood typing is, how we do it, and uh, what are the factors that we use to determine the blood type of an individual. So within blood typing, one of the things that we need to be very clear about is there's about 26 different blood groups. And those 26 different blood groups um, define our blood and can each determine really a different blood type that we have. The reason why we don't use all 26 different blood types or blood groups to determine a blood type is because they're not all reactive, or they're not all reactive enough to be able to distinguish one from the other. But we do have two blood groups that are highly reactive, enough to where we can distinguish one blood type from the next. And those two groups are the ABO blood group and the RH blood group. And so these are the two blood groups that we will go ahead and focus our look at during this video here, which I think will be a little bit shorter of a video for us. Understand that every cell that we have, our red blood cells, are no different. They've got antigens that are found on the surface of a cell, all right? And the antigens that are found on the surface of a red blood cell or a erythrocyte are called agglutinogens. And these agglutinogens define the characteristic or the blood type that is associated with those red blood cells. And this is what we typically define those agglutinogens, or what we define as the ABO blood group. And so you have A antigens, you have B antigens. They're the only antigens that we have. They're the only two antigens that we have. You have A antigens or B antigens. People that are type O blood actually lack antigens. There are no antigens on the surface of their red blood cells. And so we only have two agglutinogens that we're dealing with, A and B. And those A and B antigens are what are responsible for the production of type A blood, type AB blood. This is a codominant trait. All right, so both of these traits, both of these proteins are in control, if you would, or are expressed. All right, you can have type B blood. And again, if you are type O, that means that you lack any antigens that might be on the surface of the red blood cell. Now, every or almost all of our red blood cells also then produce an antibody or an antibody, something that is going to attack the other agglutinogen. And these antibodies are what we call agglutinins. So if you are type A blood type, the opposite of that would be type B, and so you would actually produce B agglutinins or anti-B antibodies. If you are type B blood, then you would produce A antibodies or A agglutinins. And so if you're type B blood, you can't take type A blood. So if you get type A blood in you, your A antibodies attack it. They clump it. They call coagulation. If you're type A and you're given type B blood, then your B antibodies would attack the B blood that was given to you and cause coagulation. If you are type AB blood, you don't have any agglutinins because you have both A antigens and B antigens. So there's no need to have any kind of antibody because you've got both antigens present on your surface. And if you're type O blood, that means you, once again, don't have any antigens. You have no agglutinogens, but you do produce both A and B agglutinins or antibodies. And so this is why people that are type O can give to anybody because they lack the antigens, but they can only accept O blood because they produce both A and B antibodies. People that are type AB have both agglutinogens, they have both antigens on their surface, so they don't produce any antibodies, so they can accept from anybody. That's why they are the universal recipient. All right? Type O is the universal donor. Right? And again, people that are type B blood can only take B blood because they produce A antibodies. And people that are type A can only take A blood because they produce B antibodies. And that's exactly what you're seeing here on this slide. And so, once again, just to make sure that it is perfectly clear, let's do magenta. All right, type O individuals. These guys here are the universal donors. All right, because they can give to anybody because they lack agglutinogens. All right, but they can only take O because they produce both A and B antibodies. All right. Type AB individuals, again, are the universal recipients. Because they have both A and B agglutinogens or antigens, but they don't produce any antibodies. So they can take A blood, they can take B blood, they can take AB blood, and they can take O blood because O, again, has no antigens, no agglutinogens. So AB people, y'all can freeload off of whoever you want. You can take, but AB blood can only give to AB people. Right? You can only give to AB people because if someone who is AB, if someone who is A receives AB blood, B antigens are on there. And we know that people that are type A produce B agglutinins or B antibodies. And so these antibodies would actually attack the B protein on the AB blood. Right? Likewise, people that are B can only take B blood because if they took AB blood, they're producing A antibodies that would attack the A antigen that is on the surface of those blood cells. And so B is B only, and A is A only. I also want to just draw your attention here to the genotype. I don't know if this is something that stood out to you or not in the lab component, but when we write out genetically using the genotype, each of these proteins are actually carried on an immunoglobulin protein, and so this A antigen would be carried on an immunoglobulin, and maybe this one came from dad. And then this A immunoglobulin maybe came from mom. Right? And that's how you get your blood type. You get one allele from dad and one allele from mom. Well, there's a couple different ways in which you can be A blood. So you can get an A if mom and dad are both A, right? Or if mom and dad are both AB, or if mom is A and dad is AB maybe. You could end up with a person that is A. Right? But let's say dad is A blood, AA, but mom is Maybe mom is B, but her second allele is actually an O, and actually that's not the right way to write that. It would be like this, right? Little I, because it's a placeholder. Right? So maybe mom looks like this. All right? Just ignore that. Uh, Big B with an O. All right? Well, if that's the case, dad is going to give the A, and if mom gives the O, you could still have a child that's A blood. Right? So that's what that AO means. That O just means that that second allele came from a parent who at least was a carrier for O blood. Right? Mom is not O, mom is B, even though she's carrying in an O. Right? Same thing here. Right? Big BB, right? If you're what we call homozygous for B, all right, that would be immunoglobulin B, immunoglobulin B. We got one B allele from mom and one B allele from dad, or you could be B O. Right? So one of those alleles would have had to have been from a parent who was at least a carrier for O blood. All right. Now, if that person was O, so let's say dad was, well, let's say mom was B blood, all right, and let's say dad was O. All right, then in that scenario, that child would be BO. Right? For AB blood, you've only got one choice. One of those immunoglobulins is carrying an A, and one of those immunoglobulins is carrying a B. Same thing right here for O. All right. This is me. All right. I am type O. In fact, I am what we call O positive. So I can only give blood to individuals that have an Rh factor of being positive. So I can give to A positive, B positive, AB positive, and O positive people. I cannot give blood to somebody who is A negative, B negative, AB negative, or O negative. And the reason for that is because the RH group, right, the RH group is the second reactive group that we use to define blood type. And so there are three antigens that are associated with RH, the RH blood group, C, D, and E. And antigens C and E are negative, but if you have the D antigen, then that is positive. And so if you are AB positive, that means you have the D antigen. But if you're AB negative, that means you either have the C or the E antigen that is present. All right, and that's exactly what you see here. By the way, RH, all right, that actually stands for rhesus because it was discovered within the rhesus monkey. And so that blood group was first identified within the rhesus monkey, and therefore we call it the RH blood group. Now, the interesting thing with this whole scenario is if you are RH negative, you can give to anybody that is RH negative or RH positive because that D antigen is not reactive. It's not going to cause a reaction in those individuals. If you are RH positive, you can only give to an individual that is RH positive. If an RH negative person receives blood that is RH positive, so if a Rh negative person receives blood that is Rh positive, it causes that Rh negative individual to create positive antibodies. The first exposure is not a big deal. It's okay. But the second exposure to that Rh positive blood will then the Rh negative individual will actually attack the blood and cause clumping or coagulation within those individuals. Now, usually this is not an issue, but this is, again, why me as an O positive person, I can only give blood to A positive, B positive, AB positive, and O positive individuals. Because if my blood was given to somebody that was A negative, I would cause that person to create positive antibodies, right? And then upon the second exposure, um, then those antibodies would go ahead and attack that blood and cause coagulation within that individual. And so that's not something that we would want. This is particularly of interest when we deal with pregnancies. All right, so if you have an Rh positive fetus, if you have an Rh positive fetus, Rh positive fetus, who during the birthing process, whose blood mixes with the mother who may be Rh negative, this becomes problematic. The Rh negative mother will go ahead and develop A antibodies within her blood after being exposed to the baby's blood during the birthing process. During development, during the pregnancy, mom's blood does not cross the placenta. And so it's not like the mom's blood is mixing with the baby's blood or vice versa during pregnancy. The placenta and the placenta keeps that separate. There's what we call a blood placenta barrier that separates the two blood. All right, so it's not an issue during the pregnancy. It's only an issue during delivery. There's a lot of ripping and tearing and bleeding and things like that that goes on, and it's possible for the blood to mix. Upon the second pregnancy, if the fetus is positive and mom is negative, the antibodies can cross the placental barrier. And the A antibodies will then attack the fetus's blood that is Rh positive. If that happens, that fetus will not necessarily be aborted. Pregnancy won't be terminated. Chances are the fetus will develop and grow, and a normal birthing process will happen. But that baby will now be diagnosed with hemolytic disease of the newborn, or erythroblastosis fetalis, which is the other name for hemolytic disease of the newborn. I'll write that out for you. Erythroblastosis fetalis. Right. Basically, destroying the red blood cells within a fetus is what erythroblastosis stands for. It's the same thing as hemolytic disease of the newborn. Chances are that child will be anemic his entire life, because that's what would happen. Those because of the antibodies, the positive antibodies attacking the red blood cells and the positive antigen during fetal development, those red blood cells will forever be damaged, and that child will more than likely be anemic for their entire life. And so to prevent that, we'll go ahead and commercially, it's what we call ROGAM, but it's an Rh blocker is what it is. And so what it does is it basically, we give this to the mother, and the ROGAM will cover the mother's red blood cells, blocking the negative Rh factor. And so if the baby's blood does mix, it's almost like the Rh negative factor of the mother can't recognize the positive Rh factor within the fetus. And so there's no There's no issue with that. And so that is blood typing. Um, we know that within the lab, we always expose a patient's blood to each of the antibodies. All right, and the reason why we're exposing it to the antibodies is in case, or to see for, to check for a reaction. If it coagulates, then we know that whatever the antibody we put in, that's the blood type of the individual. So if a if you're in the A well and you add your blood in and you add in anti-A and it coagulates, then you know that that blood contains A antigens. Right? If you're in the B well and you add B antibodies and the blood coagulates in there, then you know that that blood contains B antigens. Right? Same thing with Rh positive. Right? If you add the blood to the Rh positive well and you add your Rh positive antibodies in there and it coagulates, then you know that that blood has has positive antigens that are in there. No coagulation means that those antigens are not present. Right? And so if there's no coagulation in A or B, that means there's no antigens that are present, which means it's type O. And so that's kind of a review of um, the blood typing lab that you would have done in lab. So with that, I'm going to call this a wrap. You've now done all of the lectures that you need to for all the information for blood. All right, and I ask that you continue to review, continue to ask questions, and I'll catch you on the flip side.